Let's continue our discussion of molecular biology in chapter 2.4 on proteins. So the first thing we need to understand about proteins is that they are made from building blocks called amino acids. And I know you're probably so tired of drawing these by now, but if that's the case, then you should be able to do this in your sleep. So we always start off with a central carbon atom, which is bonded to a carboxyl group. And a carboxyl group is COOH. Okay? On the other end of this central carbon atom is an amine group. Amine is NH2. Amino acids have an R group, and then we fill in carbon's fourth bond with a hydrogen. Now, the hydrogen can be up here and the R group down here. This can be on this side. Doesn't matter. We have to remember that molecules are three-dimensional. They can move around. Okay, but the arrangement is important. All right, so let's practice how to make a dipeptide, or how to join two amino acids. So first of all, you're going to need to join or to draw two amino acids. So here's one, central carbon, carboxyl group, amine group, R group, and a hydrogen atom. Same thing over here. Central carbon atom, carboxyl group, amine group, R group, and hydrogen atom. All right. Now, here's what we're going to do. We're going to combine this hydrogen atom with this hydroxyl group, okay? And that's going to free up nitrogen and carbon in such a way. So nitrogen's supposed to make 3 bonds, and if we steal this hydrogen, it's only making 1, 2 bonds. Carbon is supposed to be making 4 bonds. If we steal this hydroxyl group, it's only making 1, 2, 3 bonds. So that's going to leave us with the opportunity to bond this carbon and that nitrogen. Okay, so afterwards, that's going to look something like this. Central carbon atom, carboxyl group, but I'm not going to draw in this OH group. I'm just going to leave that blank for a minute. Nitrogen, that amine group is still intact, so I didn't do anything with this over here. I didn't do anything with the R group or the hydrogen atom. Over here, I have my central carbon atom. I did not do anything to this carboxyl group, so that stays the same. Here's my amine. Okay, I did not change this hydrogen, so I'm going to leave that alone for a minute. Okay, here's my R group and my hydrogen atom. So, again, carbon needs to form 4 bonds. 1, 2, 3, and then 4. Nitrogen needs to form 3 bonds. 1, 2, and then there's the third one. And of course, we need to add in that we removed water. And that water is coming from right here, H2O. Don't forget to include that in your drawing. Okay, so we've got a total of about 20 amino acids that are used to make polypeptides, or proteins, in almost every single living organism. We all use the same 20. And again, they all have our carboxyl group, an amine group, and they have this R group, okay? Now, what makes one amino acid different from each other is the R group. They all have this other stuff. That's exactly the same, but the R groups are different. And you can see down here that the R groups are quite different. Some of them are just a hydrogen atom, so it would be a hydrogen atom up here. Some of these are these crazy complex structures sticking up off of here. Okay, those are our R groups. Now, once I stick a bunch of amino acids that are all differing in their R groups together, I get what's called a polypeptide. Peptide referring to that bond between the carbon and nitrogen atoms of 2 different amino acids, that peptide bond, poly meaning many. So when I stick a bunch of amino acids together, I get a polypeptide. And different polypeptides can differ in a couple other ways. Okay, first of all, they can be different lengths. Some amino acid chains or polypeptides are quite short and some are quite long. They can also differ in the number of amino acids, or sorry, the type of amino acids that are present. So maybe some polypeptides are going to include this one, this one, this one, this one, and this one. And another polypeptide is going to include this one, this one, this one, this one, this one, and this one. Okay, so different combinations there. And the other thing that can differ is the order of the amino acids. So putting them into a different pattern is going to give each polypeptide a very unique property. So how does this fit in with the rest of your cell? Okay, well, don't forget that this is part of a process of a living thing. So there are other encounters in the cellular organelle world involved. So here's the basic steps for how proteins are generated. Well, first of all, we start with DNA. In eukaryotes, we're in the nucleus. Okay, and that DNA contains the codes for making the proteins. So the DNA has the instructions. The DNA is a lazy boss. Okay, it says what to do, but it never leaves the nucleus. So what happens is those DNA codes are transcribed onto mRNA. Don't worry if you don't know what this word means just yet. We're going to talk a lot about that later. Just write it down for now. So those codes are kind of rewritten onto a strand of mRNA. That mRNA leaves through one of the nuclear pores and it heads to a ribosome. That ribosome is going to translate the mRNA codes into a particular polypeptide sequence. So each of these codes, again, more on this later, is going to cause a different amino acid to be added to the polypeptide chain. Where this happens is determined by what the protein is for. So if the protein is supposed to stay in the cell, it's going to be done by one of the ribosomes that's free floating in the cytoplasm. Okay, so if we're staying in the cell, it's just going to be done by a ribosome that's just hanging out in the cell, doesn't need to go anywhere. If it's supposed to be shipped out of the cell, like let's say you're a pancreatic cell and you're trying to produce insulin, you don't need the insulin in the pancreas, you need it other places. You got to get it out of that cell. Well, that's going to be done by a ribosome on the rough ER, because the rough ER is then going to hand that off to the Golgi. The Golgi is going to wrap that up in a vesicle, and then it's going to ship that sucker out of the cell via exocytosis. So a lot of times, understanding where a protein is synthesized, okay, that's going to tell us a lot about what that protein is used for. Okay, so for a lot of reasons that we're going to get into throughout the year, proteins are really the workers of our cell. They're controlling a lot of our cellular reactions and therefore controlling the expressions of a lot of our traits. How does that work? Well, remember, cell differentiation looks something like this down here. Okay, all cells possess all genes. Why is a liver cell different from a skin cell? Well, because in a liver cell genes or liver genes are turned on and skin cell genes are turned off. Okay, so what does that mean on a protein level? Well, that means that proteins that are needed for that type of cell are going to be made, and proteins that are not needed for that type of cell are not going to be made. And that's what we call gene expression, when cells only make the kind of protein that they need in order to be that type of differentiated cells. Some proteins are common to all cell types, okay? So like let's say something having to do with like manufacturing cellular organelles or something involved with cell division, like something that every cell has to do. I would expect that gene to be found in every cell and for that protein that's made from the gene to be found in every cell. If it's something that's really specific though, like again, the ability to produce insulin, well, only your pancreatic cells are going to have those insulin producing genes turned on, okay, and produce that insulin protein. You don't need your skin producing insulin, so those genes would be turned off. So if you've been paying attention so far, you should be able to answer these few questions. What type of reaction links amino acids together? Okay, so that's a great example of a condensation reaction that produces water. And this occurs on the ribosome. Okay, so again, different ribosomes produce proteins destined for different places. This creates a peptide bond. Okay, and it also produces a dipeptide if there's only two, but I'm more than likely putting a bunch of them together, and so this is called a polypeptide. Since I'm building things, that is anabolic, so it's going to require energy and form bonds. If you have no idea what I'm talking about here, you should probably really reconsider what's going on in your academic life. This should be a review from 2.1 and so far what we've talked about in 2.4. So if you're doing these chapters a little bit out of order, make sure that even if you don't understand this now, that you get this stuff some other time. Okay, now you're going to hear me say this about a gajillion times, that proteins can function like locks and keys, right? And locks and keys are highly dependent on their shape. It's the reason why my door key only works for my door and not like LeBron James' palace. So different keys have different shapes. They do different things. Same with proteins. So what determines a protein's shape? Well, protein shape is determined by amino acid sequence. We'll talk more about that later. But what I want you to know for now is that the function is also determined by the shape. Okay, so if we change that shape, we're going to change or disable that function, and that can mean some pretty bad things. So keep in mind that proteins are supposed to be a very specific shape, and that's really important to their function. Now, proteins only achieve that specific shape, okay, when they reach their mature level of structure. And there are 4 levels of protein structure. The first one is primary, and sometimes you may see it written like that. And this is just a sequence of amino acids, okay? These are held together with the peptide bonds. And what's interesting about this is that this is what determines the other 3 levels. So the order in which these amino acids are placed determines the shape that it's going to fold into. All right, this chain of amino acids may fold into a secondary structure like one of these. So secondary is sometimes written like this. There are 2 types of secondary structure. Alpha helix, which you'll sometimes see written like this. That's a symbol for alpha, the lowercase symbol. Or beta pleated sheets. So beta, kind of something like that. Beta pleated sheets. Okay, and they're held together with hydrogen bonds. So hydrogen bonds are interactions between the partial positive charge of one molecule and the partial negative charge of another molecule. Well, those carboxyl groups that are on our amino acids have the charges that I'm talking about. So what's happening here is that there's a partial positive charge being very attracted to a partial negative charge, and it's taking, let's say they were up here originally, it's taking those positive and negative ends and it's bending them towards each other because they're so highly attracted. And that's going to bend them into these shapes. Some proteins stop here. Some are folded even further into what we call a tertiary structure, sometimes written like this. So this is when we take those alpha helixes and those beta pleated sheets and we crumple them all together. So you can even see in this tertiary structure, here's some beta pleated sheets and some alpha helices, and we took them and we kind of bent them all together and crumbled them all up. This happens because of interactions between the R groups of the different amino acids. So whereas secondary dealt with hydrogen bonds, this is when those different R groups start interacting. So let's say over here, okay, there's an R group that is really highly attracted to an R group over here. Well, that's going to bend this whole thing inwards. And that's when we get a situation kind of like this, where this part and this part start to come together. Let's say there's an R group over here that, because of its chemical makeup, really hates an R group over this way. It's going to try to get as far away from that R group as possible. They don't like each other. Okay, so that's when you're going to get something like what's happening here and here. So they're actually repelling each other. And then finally we get to quaternary structure. Again, some proteins stop here. Not everything moves on to the next one. Okay, but the proteins that do possess a quaternary structure, sometimes written as this, and this is when 2 or more polypeptides come together. Okay, so you don't necessarily have to worry about what kind of bonding that is there. You just need to know that it's one polypeptide, okay, kind of combining or bonding to another type of polypeptide. Okay, so let's list some common examples of different proteins that have different levels of structure. So spider silk, which I'm sure you're familiar with, has a secondary structure. So it is either in an alpha helix, okay, or a beta pleated sheet. And of course that's used for making spider webs. Just because it's a secondary structure doesn't mean it's not awesome. That stuff is super strong and amazing, okay? Secondary structure is just fine for that. It's fully functional without having to move past this structure. All right, insulin is an example of tertiary. So I'm going to go ahead and write the symbols and the words. You should do the same thing. So secondary, and this is tertiary. And now I want you to do something, and I want you to go back to that other table that you were working on. And I want you to write down the other word that we have for tertiary, which is globular. I know that sounds kind of dumb and like not sciencey. Trust me, it's a legit word. Okay, globular, okay, all crumpled up. Well, human insulin is an example of a tertiary structure. So those alpha helices and beta pleated sheets kind of got all crumpled together, and that protein is perfectly functional in that tertiary form. Then we have collagen. Collagen is in this quaternary structure, okay, meaning it's made up of several polypeptides, and it's part of the thing that makes up your connective tissue. So like in your skin, in your gums, okay, you have all this collagen, and it's really important to a lot of other physiological processes, which we'll get to later on in the year. But for now, we need to know it's a quaternary structure, so it's made up of several polypeptides. Now, you're going to notice that I'm going to be pretty dang picky in regards to using these words protein and polypeptide correctly. When you took your very first biology class back when you were little and cute and not as smart, your teacher probably told you that those words mean the same thing. And that's okay. All right, that's not entirely false, but now that we understand a lot more, okay, we can really start to see the differences in those two terms. A polypeptide is just a chain of amino acids. Okay, it's the sequence of amino acids as coded for in your DNA. Something is not a protein until its polypeptide chain has been folded into the correct shape and it's ready to carry out its function. Okay, so if we know that insulin needs to be in a tertiary structure, then we say this is the polypeptide that will become insulin, but it's not actually insulin until it forms its correct shape and is ready to perform its function. And that's when we can start calling it a protein. Okay? Now, again, things that require more than one polypeptide for its correct shape, okay, those are our quaternary proteins. And they come from kind of a tricky place, right? So here's a polypeptide chain, and this is part of a molecule called hemoglobin. Hemoglobin is a quaternary protein. It's found in your red blood cells, and it's responsible for carrying oxygen atoms. It has 4 polypeptides. So this has a quaternary structure, okay? And since it has 4 polypeptides, it's going to require 4 different genes. You see, each gene contains codes for making 1 polypeptide. Remember, genes are segments of your DNA. So one gene, which is one segment of DNA, makes 1 of these polypeptides. I would need another gene to make this polypeptide, another gene to make this polypeptide, and a whole nother gene to make this polypeptide, okay? So one gene, one polypeptide. That means that quaternary proteins are always going to require more than one gene so that they can get those chains of amino acids into the final shape that they need to be. Okay, so speaking of genes, let's go back and talk about a word that I'm sure you've heard of before, and that's genome. So a genome is a collection of all the genes in your DNA, and you have tens of thousands of genes, okay, that make different proteins and can do different cool things. Now, each genome is unique to that individual, okay? So that has a lot to do with sexual reproduction and the formation of gametes, mutations, all kinds of things, okay? But everybody's genome is different. Now, since those genes are code for proteins, that also means that each individual has a unique set of proteins, and we call those protein sets proteomes. So genomes are for genes. A proteome is someone's unique collection of proteins. Okay, now I said before that proteins are highly dependent on their shape, and they are. That's totally true and legit, okay? That shape is determined by a lot of different things, by the sequence of amino acids, okay, by the hydrogen bonds and their secondary structure, okay, and by the R group interactions in their tertiary structure. If you do anything to screw this up, if you're altering that structure in any way, that protein may not be able to carry out its original function, and that's what we call denaturation. And this is a permanent process. When proteins permanently lose their specific shape due to a change in their environment, we call that denaturation. So we must be doing something to alter some of these factors that go into making the protein its own awesome, wonderful shape. 2 of the environmental factors that can cause denaturation are temperature and pH. So let's talk about temperature first, okay? And here we're going to limit our discussion to high temperatures because cold temperatures don't really do much in terms of denaturing proteins, okay? But high temperatures can do some really gnarly things to these proteins. Now, it's important to note that the covalent bonds that hold the amino acids together stay intact. You cannot really destroy these covalent bonds by adding heat. However, the hydrogen bonds that hold together our secondary structure, so they're holding together these alpha helices or the beta pleated sheets, come undone when you expose them to high temperatures. And what that's going to do, that's going to kind of unwind them, and you can't put them back together. Well, if our protein needed to be this specific shape in order to do its job, and now it's this specific shape, it's not going to be functional anymore. It's not going to be able to catalyze those reactions. Okay, so let's take a look at different temperatures and how they can affect enzyme activity. And we're going to annotate this graph. So let me give you just a second to kind of jot that down. You don't necessarily need the units. The units don't matter. I'll tell you why in a minute. Okay, but what's going on with your X and Y axis is definitely important. we can see for this enzyme, the maximum activity or the maximum productivity happens right here. And this is what we call the optimal temperature. Optimal meaning most awesome. Okay, that's the temperature at which this enzyme is the most busy, the most active. Okay, and that's going to be at the peak of our graph. Down here in this area of our graph, we can see that at colder temperatures, the enzyme activity is going down, or the reaction rate is going down. That has nothing to do with the enzyme, okay? This has everything to do with cold temperatures slowing down molecular collisions. So cold temperatures slow down molecules. Okay, meaning less reactions. So low temperatures tend to make slow reactions. Okay, things just slow down in these cold temperatures because molecules aren't moving fast enough, they're not colliding as much, they're not colliding with enough energy, all sorts of things. Then after this optimal temperature, we see this crazy sudden drop off, like from 100% activity down to 0 in like not very much time. Well, this is where our enzyme is being denatured. Okay, so after the optimal temperature, if you keep increasing the temperature, okay, what you're going to do is you're going to denature that enzyme, you're going to unfold those proteins, and now that enzyme isn't able to catalyze those reactions anymore because you done messed with its shape. We're going to see a similar phenomenon for pH, but for a different reason, okay? So the end result is the same, whereas before we had a nicely folded structure, that's going to come undone. It's going to come unwound. But this time it's not because temperatures are breaking hydrogen bonds. This time you're kind of adding in blockers between those hydrogen bonds. The hydrogen ions or hydroxide ions produced by acids and bases will sometimes get in the way of those hydrogen bonds, and they're not going to be highly attracted to each other anymore. So remember that this shape is determined by the positive and negative charges on these amino acids being highly attracted to each other. Well, if I put an ion, like a hydrogen ion or hydroxide ion in between them, that attraction no longer exists and it's going to unwind. Okay, again, this is a permanent change to your enzyme shape. Okay, so if we have to trace what's going on with the effect of pH on enzymes, we're going to see a really similar phenomenon. Okay, so here we've got pH on this part of our graph, and we've got rate of reaction or maximum activity. Those all mean the same thing. Okay, and we're going to get something that looks like this, kind of like a nice cute bell curve. Up at the top here, okay, we've got our optimal pH. Okay, and our optimal pH is when is the pH of our environment when our enzyme is working the best. Down here on this end, lower pHs, if I lower the pH of something, that's acidic. Okay, so here the hydrogen ions from acids are denaturing the proteins. Okay, so those denature proteins. Okay, over here it would be the hydroxide ions from different bases that denature the proteins. Okay, now again, it's important to note that different proteins are going to have different optimal pHs. Some proteins like their pH to be up here on more of the basic ends, like some of the proteins found in your blood. Some of them like to be down here in the acidic end, like some of the proteins found in your stomach. Okay, but It doesn't matter. They all have the same kind of thought here, an optimal pH. Okay, and then on either side, it's either too acidic or too basic for them to maintain their functional shape. All right, so I want to go back and amend something that I talked about earlier. I know you're not used to me making mistakes, okay? This wasn't really a mistake, but it's something that we often talk about. We always say, how many amino acids are there? And you, because you're wonderful and a good student, you say 20. Well, yes and no. Okay, almost every organism on Earth uses these same 20 amino acids. But because this is biology, there's of course an exception to that rule. There are a very small number of organisms that actually use 22 amino acids. Okay, these two are weird and not very common, but they are used in the polypeptide chains of just a couple organisms. Okay, and that'll do it for chapter 2.4 on proteins.